So, what is your blood type? This is one of the first questions you're going to ask any trauma patient, and it's a vital piece of data before any surgery, and it's a cornerstone of any safe blood transfusion. As a health sciences student, you really know what makes blood A positive or O negative or what have you, right? So today I'm going to move beyond the basics. We're going to master the science of blood typing, specifically the 8 common types that you see every day. But we'll also look at why the system is actually far more complex than that. Welcome to your essential guide to human blood types. First of all, to understand the blood types, you have to understand 2 key players, and that's antigens and antibodies, right? So antigens are proteins or sugars sitting on the surface of your red blood cells. Think of them as your cell's ID card. They tell the immune system, hey, I belong here. I'm supposed to be here. I'm not an invader. Antibodies are proteins circulating in your plasma. They're kind of like the security guards. If they spot an antigen that they don't recognize, they're going to attack it, right? And so this leads us to the most widely known blood typing system, and that's ABO. The ABO system is determined by which antigens that you inherit. If you're type A, such as myself, you have A antigens on your red cells. Consequently, your plasma contains anti-B antibodies. So if I ever receive type B blood, these antibodies will cause a massive reaction and it could potentially kill me. So that would be a bad thing. Conversely, if you have type B blood, you have B antigens and anti-A antibodies in your plasma. So if I were to give you my type A blood, that would cause a similar adverse reaction, then I could kill you without even thinking about it. But anyway, so that would be bad. Things get a little interesting if you're type AB. And so here you have type A and B antigens. So crucially, your plasma has no anti-A or anti-B antibodies. So because it doesn't have the guards to attack A and B, that way type AB is known as the universal recipient for red blood cell transfusions. So you can receive anybody's blood. And finally, we have type O. And so these red blood cells have neither A nor B antigens. They're essentially naked. However, the plasma contains both anti-A and anti-B antibodies. So because cells lack antigens to attack, type O is referred to as the universal donor. These people can give blood to literally anybody. And so type A and B blood antigens are terminal carbohydrate structures that are attached to H antigens on red blood cells. Type A antigens are defined by the terminal sugar N-acetylgalactosamine, and type B antigens are defined by the terminal sugar D-galactose. Both are added to a common H antigen precursor. But A, B, and O are only half the story. You also need to know your Rh factor. This is also called the D antigen. This is simpler than ABO. You can either have the Rh antigen or you don't. If you have it, you're Rh positive. And that's about 85% of the human population, which is Rh positive. If you lack the Rh antigen, you're Rh negative. Unlike the ABO system, Rh negative individuals do not naturally have anti-Rh antibodies. They only develop them if they're exposed to Rh positive blood. That could be either through an incorrect transfusion or during pregnancy. And a lot of times during pregnancy, this was a major issue back in like the 40s and 50s before we kind of learned more about this system. When we combine ABO and Rh, we get 8 common blood types that you must memorize. So for example, O positive blood means that you have O antigen, you don't have any antigens, right? And you have Rh antigen, right? So you don't have A or B, but you have Rh. You also have anti-A and anti-B antibodies. A minus means you have A antigen, but no Rh antigen. And so you have anti-B antibodies, and you have the ability to make anti-Rh because you don't have the Rh factor. So here's your table, and that's your Bible essentially for rotation, right? So why don't you take a screen capture of this now, make sure you memorize this. Understanding who has which antigens and antibodies dictates compatibility. So why does this match matter so much? If you give the wrong type, the recipient's antibodies attack the donor cells. This causes a reaction called agglutination, and that's a rapid clumping of red blood cells. These clumps will block blood vessels, and the immune system destroys the donor cells, releasing massive amounts of hemoglobin into the bloodstream. And this leads to an acute hemolytic transfusion reaction, which is a medical emergency that can cause kidney failure, shock, and death within minutes. So don't give the wrong blood. Now, everything we just discussed is critical, and it covers about 99% of your daily clinical life. But here's the secret that's going to make you a smarter provider. Humans do not have just 8 blood types. There are over 350 recognized blood antigens, and they are organized into over 40 distinct systems. The AB and Rh systems are the most immunogenic, and that means that they cause the strongest reaction. But systems like Kell, Duffy, Kidd, MNS, and Lewis are also present on red cells. The origin of the 350 plus known human blood types is a fascinating story of genetic mutation, geographic isolation, and evolutionary survival. While we focus on the AB and Rh systems in a clinical setting because they trigger the strongest immune responses, these other minor blood groups are the result of millions of years of human adaptation. The most significant reason we have so many blood types is natural selection. Blood group antigens on the surface of red blood cells often serve as docking stations or gateways for bacteria, viruses, and parasites. For example, the Duffy blood group is a classic example. So certain populations in West Africa evolved to be Duffy negative because the Plasmodium vivax malaria parasite uses the Duffy antigen to enter red blood cells. By turning off that antigen, those populations gained a distinct survival advantage. There's also evidence that people with type O blood are more susceptible to severe cholera. And this might explain why type O is less frequent in regions where cholera was historically endemic, such as in the Ganges Delta in India. So some pathogens mimic blood antigens to hide from the immune system. Humans evolved new, varied antigens to stay one step ahead of all of these evolving threats. At the molecular level, a blood type is simply a specific variation in a gene that codes for a protein or an enzyme on the red blood cell membrane. Some of these blood types are caused by single nucleotide polymorphisms, which we also call SNPs, and that's just a single typo in the DNA sequence. A single amino acid swap can change the shape of a surface protein just enough that the immune system recognizes it as a different type. So some rare types occur also as a result of gene deletions. And that's when an entire gene is deleted or silenced. This leads to a null phenotype, such as in the Bombay phenotype or Rh null. Before the advent of global travel, human populations lived in relatively isolated pockets for thousands of years. And this led to something called the founder effect. If a small group of people migrated to a new area, like the Americas or remote islands in the South Pacific, one person had a rare mutation, and that mutation could become the dominant type in that specific population over generations. So, for example, this is why certain indigenous populations in Central and South America are nearly 100% type O, while the B allele for type B blood is actually most frequent in Central Asia. And so while we call these molecules on the surface of red blood cells antigens because they trigger an immune response, these molecules aren't just there for decoration. They actually have functional roles in the body. They're actually receptors on the surface of the cell membrane, right? And so some antigens can act as channels to move water or urea or other chemicals in and out of the cell, such as in the KID system. Others act as scaffolding to help the red blood cell maintain its flexible biconcave shape, such as in the Rh system. Still others actually help cells stick to blood vessel walls when needed. So while compatibility in these systems is rarely a problem for a single transfusion, it becomes vital for patients receiving multiple transfusions over their lifetime. And so this is the case for individuals with sickle cell disease or thalassemia. The presence or absence of these minor antigens can also create extremely rare blood types. For instance, in the Bombay blood group, which I previously mentioned, these are individuals that cannot even make the basic base structure needed for ABO antigens. While they appear as type O on standard tests, they can only receive blood from other Bombay individuals. And so you have to know about this, because if you encounter that in a clinical setting, you need to be aware of that, right? It's extremely rare. So here's essentially a summary. So number one, the ABO system is defined by inheriting A or B antigens. Your plasma guards against what you lack. Number two, the Rh system is inheriting the D antigen. That's plus or minus. We can combine those with the ABO, and we get A plus, B minus, etc. So we've got 8 blood types. Number three, a mismatch in blood types causes agglutination, and that's a fatal clumping of blood cells. Number four, while ABO and Rh give us 8 main types, human blood has over 350 antigens. And this complexity is something you must respect as a healthcare professional. Knowing this isn't just for passing exams, it's actually a life or death situation, and it's about keeping your future patients safe. If you found this essential guide helpful, please hit the like and subscribe button for more health science education. Go ahead and hit the little bell icon, and then you'll be notified when I post changes and updates and new videos. And go ahead and leave a comment if you have any questions or comments to add to any of this information. Leave those in the comments below. And then If you have a topic you're interested in, leave that in the comments as well. And so thanks for watching my channel, Dr. Cash Chemistry, and good luck with your next exam, and stay curious. Thank you.